Okay, maybe we are moving to the next presentation by Ms. Fiadini Putri, a presentation on the acclimatization, which was mentioned earlier by Mr. Abdurrahim. So first of all, I would like to introduce the profile of Ms. Fiadini Putri. So Ms. Fiadini Putri has more than 10 years experience in training and teaching human resources working in agriculture. She is an agronomist and horticultural specialist. She has extensive professional experience in horticulture, global trends, research, inorganic nutrients, and secondary metabolites of plants. She trains in acclimatization as part of tissue culture technology. Currently, she works at the Lembang Agricultural Training Center, the Ministry of Agriculture of the Republic of Indonesia, as lecturer. So Ms. Fiadini is currently completing a PhD in agronomy and horticulture at the Bogor Agricultural Institute. So for this session, she will deliver a presentation on the acclimatization. Ms. Fiadini, if you have a video to be played or a PowerPoint to share, please.
Okay, now is the part of the acclimatizations for banana plantlets. Okay, acclimatization is a process of adaptation or transfer the plants from the internal environment to the outside environment, from the controlled environment in the culture vessel to uncontrolled environment. And this is a final stage of micropropagations. So acclimatization is actually the physiological adaptations of the plants to change in the climate or environment, because in the tissue culture vessel or in the incubation rooms, there is a different light or temperature conditions and then in the field. So in this case, the banana plantlets are being acclimatized from the culture vessel to the greenhouse and outdoor environment. So this acclimatization is used to adjust from the high air humidity to low air humidity, improve root function to absorb nutrients from non-agar media into soil, and maximize the photosynthesis process, adapt from sterile environment into environment where microorganisms present. And the material and tools, just like you saw in the video, you need the complete plants in the tissue culture. So we need the plantlet who have complete roots, stem, and also the leaves. And also the acclimatization media, which is sand, compost, soil, and husk charcoal. We need the root hormone and foliar fertilizer, fungicide and bactericide, plastic train sprayers, and all the miscellaneous clear plastic and also autoclave. And to prepare the acclimatization media, as you see on the video, for the first acclimatization media, we need to arrange the medium, which is sand and compost. And we mix the media and put it in the clear bag or clear plastic bag. And after that, we need to sterilize it. And we cool it in the plastic tray. And also we need to sterile the media using the fungicide and bactericide and let it stand for a night or for a day. So this is the acclimatization process of the banana plantlets. We need to take the banana plantlet out from the culture bottles and we clean it up under the running tap water. And we have to make sure it's already clean. And after that, we soak into bactericide and fungicide solutions and let it dry. And after that, we need to dip the root plants into the root hormone. And after that, we plant the plantlets into the acclimatizations media. And this potting medium should be uniformly moistened with a small amount of water before it placed into a nursery containers. And this will help to station the roots during the planting. So this is I think it's compulsory to do. And after that, we put the plastic above the plastic tray. And it will be stored in the plastic lid cover with 55% to 75% of seed net for 3 to 4 weeks. And for growing the banana seedling, we need to transplant, and the transplanting is the process of moving from the acclimatized banana seedling into the polybags. This is the second phase of the acclimatizations. And the composition of the polybag media is soil and fast charcoal for 4.5. And the plantlets are removed from the acclimatization medium slowly. Make sure that you will not damage the roots. And after that, you put into the polybag. And save the seed in the polybag under the plastic lid cover, then watering them in the morning. And we store the seedling for at least 1 to 2 weeks, and the cover is labeled with the date of planting. So while the seedling inside the plastic lid cover, we need to provide them with the foliar fertilizer 2 times a week. So this is how to establish the seedling before replanting to the field. The seedling in the polybag that have been covered for 2 weeks were left under 55% shade net for 1 to 2 weeks. And the seedling that have grown new leaves are transferred into mounds or beds in the open land without shade nets, but it's still inside in the polybags, still on the polybags. And every 2 weeks, the seed will provide with the nitrogen fertilizer, about ½ teaspoon per polybag of banana seed. In here we use urea. Yes. Yes, urea is 45% of nitrogen in this nitrogen fertilizer. And if attached by a caterpillar or insect, we can spray this seedling with a 2 millimeter per liter of pesticide. So after about 2 months in the nursery, the banana seedlings are ready to transplant into the open field. So this is the key to successful weeding, to keep young sensitive plants free from any form of environmental stress. For the idea of weeding, this weeding system can be regulated to control the most important environmental factors such as light, water, temperature, humidity, and also wind. So we have to make sure that the condition is suitable for the banana plantlets to grow, because as we know, when we culture them into the culture vessel, they are very delicate, so we need to harden them before we transplant the plants into the field. Okay, I think that's all for the presentations in the acclimatization. If you have questions. Okay.
Participants, do you have questions for the presentation from Ms. Fiadini? Maybe maybe Ms. Fiadini, could you describe further details about urea that you mentioned earlier, and how it could possibly obtain in other countries, despite of differences with maybe climate or season with Indonesia?
Okay, so in Indonesia, we have a type of nitrogen fertilizer. We call it urea. It's just like, it's not like a powder, maybe. Yeah, and it only contains about 45% of pure nitrogen. So this fertilizer is commonly used by the farmer. So we can use for the acclimatization. It's enough for the acclimatization. So in Indonesia, we call it urea, but might be different from your country. But the clue is These fertilizers only contain about 45% of pure nitrogen. So this one that we use, maybe if you have a different brand of fertilizer, you can use it.
Thank you very much, Ms. Fia. So there's a question from one of participants from Indonesia about how many did the explant in the culture bottle? How is volume bottle? I'm not sure what is in the culture at explant. I don't know. I don't know the meaning of this question. Maybe in the chat, maybe you could check it, Ms. Fia.
On the chat, not on chat now. Sorry, Ms. Megan, we will use available. Okay, no.
How many did that explant in the culture bottle? Or maybe about how many is the amount of the explant in the culture bottle? Okay. And the volume of bottle required in the process of culturing explant banana. Yeah, I believe maybe that is the question.
Okay, this is how many explants in the culture bottle. Actually, in the culture bottle, we can see the amounts of the plant explants is maybe, it's a plantlet. It's about maybe a 10. And if it's too dense, you need to subculture it again. So we have to consider also the media. So if the media is already, we run out of the media and the plantlets don't have another media to consume, we need to subculture it in another bottle. So for the volume, I think it's this size is we usually it's about 20 millimeters of the media. Yeah, we can put the media about 20 milliliters. It's 20 milliliters in this culture bottle, and we can see the conditions of this plantlet. And if it's too dense, you can subculture it again. Because sometimes there is a big plantlet and the small one. Yeah, you have to make sure it's still have enough media for them.
Okay, thank you very much, Ms. Fiadini. I believe the answer helps. There is another question from Mr. Ferguson Nainggolan. How to anticipate the fungal attack before planting? Maybe he refers to the planting of the seeds obtained from the tissue culture.
Okay, to anticipate the fungal attack before planting, that's why we use a fungicide to sterilize the plantlet. And before that, we have to make sure that the plantlet is also clean when we take it from the bottle. We have to make sure it was clean, because if there's still added media on the root of the plantlets that we were taken from the bottle, it is a good media also for the fungi and the bacteria to grow. So we have to make sure when we take the plantlet outside from the culture bottle, we have to clean it, and we have to make sure there is no more added media or tissue culture media on the root. So the key from this is the sterilization and the fungicide. We use the fungicide.
Okay. Okay, thank you very much, Ms. Fia. One question. Maybe I'm wondering, just in case, maybe the case where the tubes are not in a are not well sanitized, for example, what will happen to the seeds? I mean, will the seeds will be defect, or maybe it can still grow, but maybe there will be maybe fertility or productivity issue with the plant?
Yes, this is why this is one of the most important phase of the tissue culture process. maybe we can spend several months in the incubation room, but if we are not succeeded in the acclimatization, so we will not have a seedling to plant on the field. So that's why we need to address these situations. And okay, that's why because excuse me. Oh yeah, okay, and the tools must be clean. All of the tools must be sterilized first because it's a very delicate plant. You know, when we grow a plantlet in the tissue culture, it's a very delicate. So during the in vitro, plantlets grow under special conditions with a relatively airtight cultivation vessels. The air humidity is higher, the air radiance is lower than on the field. And the use of the closed vessel, of course, we need to close it to prevent the microbial contaminants. But, you know, just like a baby, when we taking care of this plantlet and we want to plant into the field, so this is a very delicate and easily to wilt or rooten or decay, and it has a high mortality rates, including in the banana plantlets or every plant that you culture. So this is a very important phase. And you know, when we plant the tissue culture, we use the tissue culture. In the culture threshold, there is a less air turbulence, and this is increase the leaf boundary layers and limits the inflow of the CO2. So, for example, when we do the acclimatizations, as you see on the video, we use the plastic lid cover in the tray without any holes. This is because very limited inflow of CO2. And we need to gradually correction that conditions before we plant into the field. So just like a baby, we weaning them. So we have to train the plants before we move it into the field. And we need to do that gradually.
Thank you very much, Ms. Lia. So other participants, would you like to address question or feedback? Please use the Q&A feature or use the raise hand feature if you want to address your response verbally. Or maybe I could ask one question to Ms. Fiadini and maybe Ms. Sani and Mr. Abdurrahim would also answer. I'm reading several literatures which says about the advantage of using a seedling that is produced from a tissue culture method. Like for example, some seedling from a proper tissue culture procedure could result in productive plantations, or maybe result in a plantation that is very resilient against seasonal change or resilient against pest and disease. So I'm wondering, with the proper method that all of the trainers have explained earlier, what is the specialty about this seedling that is produced from this tissue culture method compared to normal seedlings? Thank you.
Okay. Okay, maybe I will try to answer. Just like I mentioned before, the tissue culture is a very useful method, and it has a lot of benefits, especially if we have a spatial character of plants. Using a tissue culture is make sure that we have the same characteristic with the mother plants. So it's a little bit different when we grow the plants from the seed, from the fruit of the plants, for example. There might be another characteristic from the conventional propagations, for example, from the flowers, from one to another, but using tissue culture is make sure that you have the exact character from your mother plants. So it's very useful if we want to, might just like, oh yes, okay. So we can use a special variety if we want to result a lot of plants with the exact variety. And once again, it's free from virus. Using a tissue culture is make sure that the seedling that we have is free from virus and also bacteria.
Okay, maybe I can add some more. On seedling with tissue culture, not depend on season.
Yes.
We can produce the seedling continuously, not depend on season. And also we can produce a lot of seedling with rapid time, fast time, and with in the room, but we have a lot of seed, one room. And also the seedling is and all the seedling homogen, same like the mother plant, same them. And maybe for the transportation of seedling is cheaper and easier than conventional seed propagation. Maybe that's all.
Okay, and it's also very beneficial if you want to produce a kind of variety that have a resilience in a drought or pest, because using the tissue culture, we can, create the media.
Yeah.
And for selection, for example, if we want to produce a plant that resilient to the drought, so we have to manipulate the media so we can select which type of variety that will be survive in the drought. And it's a really beneficial because the conventional way is quite time consuming. Using this tissue culture, we can cut the time we produce the resilient plants. And it's also very good to produce secondary metabolites.
Yeah,
also.
secondary metabolites.
So if you want to produce kinds of phenolic compound, for example, and it's only produced on the leaves of a certain plant, we can grow the plants inside this culture bottle, and we harvest the leaves that we already provide with a special treatment, for example, the drought, because a secondary metabolite is very useful for the cosmetic and also for the drugs industry, medicine industry.
Okay.
So there's a lot of benefit of using tissue culture. Yeah. And the seedling also have a lot of advantage.
Yeah.
Thank you very much, Ms. Fiadini. Okay, for participants, do you have any further questions addressed to Ms. Fiadini, or maybe also questions that is related to the previous topics? You could address it right now as well, since all of the topics are correlated from one another. Okay, so the honorable facilitators, I'm wondering, since maybe in several regions there are power supply problems, and beforehand you mentioned about like using like air conditioner for the storage period of the tissue. So I'm wondering, what would happen if there is a power outage in the laboratory, and is there an alternative other than using the air conditioner to maintain the temperature?
Gantian kalau AC bisa diganti yang lain-lain kan? Gantian. Penggunaan AC bisa diganti sama yang lain kan?
I think that was the requirement for the laboratory, tissue culture laboratory, is that we need to control the temperature and humidity inside the laboratory. This is one of the requirements. So I think it's difficult to replace with another alternatives, because we have to make sure that the plant was grow in an aseptic environment. So I think it's a compulsory for the tissue culture laboratory. That's why we need to have constant electricity also. Even though some of the farmers also have a simple laboratory.
Oh, yeah.
Right. For example, for the alternative, we're not always use the expensive LIF, yeah, laminar airflow. There is a simple type of laminar airflow. Using in our laboratory we use the laminar airflow using UV lights and blower. But there is another container that you can use for your simple laboratory. We call it NCAS.
NCAS, yeah.
It's a kind of box. Yes, it's just only box and yes, it's made from acrylic that you can do your culture work inside the container. It's usually used for orchids, if I'm not mistaken.
Yeah.
Yes.
Orchid is still be able to produce using this kind of simple laboratory of tissue culture that sometimes the farmer have this simple laboratory in their house. So you don't need a very sophisticated tissue culture laboratory for certain plants.
Facilitators, I believe there is one question from Bangladesh. What is the difference? What is the difference in fruiting behavior and fruit quality between the tissue culture plants and the normal plant? In the chat section, from Dr. Mokhtar Hussein.
Yeah. Sometimes, okay, we experience this, that the plants from the tissue culture sometimes is quicker, right? Maybe, for example, for certain plants, it needs 1 or 2 years after we plant on the field to have the fruit. But using the tissue culture, we can cut the times and they can grow faster and also produce the fruits. For example, the bananas, the normal plants that we propagate from the sucker, from the banana sucker, they need a longer time to produce the fruits than from the banana plantlets. So this is a kind of situation that the banana plantlets that we produce from the tissue culture is already complete all this phase of their phase to grow. So when we transplant into the field, they were directly ready to produce the fruit. And this is one of the benefits of the tissue culture, that the quality of the fruit is going to be exact the same with the mother plants. It might be different with the fruiting, with the fruit from normal plants that they need a generative phase, right, to propagate. And might be there's another contamination from another plant. So I think that was the different type of behavior of the tissue culture plants.
Thank you very much, Ms. Fiadini. Dr. Mokhtar, would you like to provide further response?
It's all right, ma'am. Just one point was from my side is that tissue culture plants, as you said, that will give fruiting earlier than normal sucker. And quality means that fruit quality, storage ability, I think might be higher than normal plants. Your answer is fine. I appreciate you and thank you very much.
Thank you very much, Dr. Mokhtar Hussein. Based on the explanation, I'm assuming that maybe the condition, maybe the seedling will imitate the condition of the mother plant, both in terms of the productivity or the resiliency, right, Miss Fiadini?
Yes, right.
Okay, thank you. So, other participants, would you like to address questions? Or maybe both verbally or maybe through written response, please. Okay. Okay. Or for Miss Sunny, for the facilitators, for all the facilitators, maybe each of you could provide final remarks before we are heading to the next agenda, which is the action plan. Final conclusion, maybe based on our interesting deliberation today.
Okay, thanks, Ms. Niken. Maybe I hope this tissue culture matter can, maybe we can sharing our experience in here in Indonesia to all of you participants. Maybe we can stay in touch, maybe to more discussion about the tissue culture. Maybe that's all. Thank you.
Okay, from Mr. Abdul Rahim, maybe final remarks.
Okay, about the explant management, I hope you must be try and learn about explant condition, about sterilization material, about the concentration of the sterilizer and sterilization time. Because our condition is different. One more, you can try and learn, and you can practice in your country, and I hope I get the success, your success in your country. Thank you.
Okay, thank you. So final remarks from Ms. Fia.
Okay, thank you, Ms. Nikan. Tissue culture is a very good method, and we can improve it in any kinds of method that we need. And I know that some of the participants already have a good experience in tissue culture. And just like Ms. Sunny said, that we hope that we still can in touch each other. We have the WhatsApp group and also the LMS system that we can share the experience from every country. I think we can't wait to hear about your experience about tissue culture in your country, and it's a very good opportunity for us to can share and also maybe discuss further about this. And we really appreciate for the committee and also from the NAM Center and also from the Ministry of Agriculture and also for the committee. And I think that's all. Thank you very much.
Thank you very much.